I'm going to talk to you today about how we can understand how plant communities, Antarctic plant communities, are responding to climate change. And I'm Sharon Robinson, and I'm one of the Deputy Directors for SAEF. My role will be in science implementation, and I'm also the Executive Director for the UOW, University of Wollongong Global Challenges Programme. And I'm delighted to be in SAEF because after having worked in Antarctica since 1996, the possibilities that we have within SAEF in terms of working across climate, biodiversity and conservation are really thrilling. And so that interdisciplinary nature that we have within SAEF is really one of the things that I'm very excited about. And in this first slide, what I'd like to try and do is show you the way in which the areas of my research come together with those other areas across, those other themes across SAEF. So my work is really in theme 2. I'm interested in the climate effects on biodiversity. And a lot of that has been around direct effects such as the effects of UVB radiation or the effects of temperature on plant communities and plant health in Antarctica. But I'm also interested in how the climate effects influence what gets into Antarctica. So wind patterns and wind strength and ocean currents can be one of the things that actually helps to bring or exclude species from Antarctica. So that's true for both native species, how did native species get there, but also how have introductions, how will future and current introductions, how do they get to Antarctica? So climate's really important in describing where the biodiversity is and how it got to Antarctica. And in my research, I'm really interested in how do we understand how the climate is driving biodiversity. And to do that, we really need to be able to scale those climate models, so data that we have on the size of kilometre pixels of weather and climate. How do we scale those to the level that's appropriate for the biodiversity? So not to scale in the middle. I've got a penguin, and the penguins, maybe a metre squared is a good space for a penguin in terms of the habitat that it's surviving in. But then we've got really, really tiny organisms, and some of the ones that I work on are mosses, where centimetres are really important in terms of the microclimate that they're experiencing. And so how do we scale from those climate models down to the appropriate scales for that biodiversity? And what sort of uncertainty does that bring in? And this is where all the statistical expertise that we have will really come in terms of understanding, can we actually do that? Can we model it? And then can we understand how much uncertainty there is in that process? And then the other area is, what does that mean for the future of Antarctic biodiversity? So what does that mean in terms of preserving the biodiversity in Antarctica? And the other thing that I'm interested in is, if we find out that a certain species is being affected by, say, drying, is there anything we can do about that? So the theme 3 to me is coming in terms of, what does that mean for the future of biodiversity, and what does that mean for, is there anything we can do about it? But I'm also interested in the way in which theme 3 and theme 2 can potentially lead back into theme 1. And so what I'm hoping that we can achieve through SAEF, and I'm really confident that we will be able to do this, is that we'll be able to look at using methodologies, the methodologies that would come up in theme 3 to inform better management and potentially perhaps alter environments to preserve biodiversity, but then also, what can the biodiversity tell us about climate? And I'll come back to some of those themes as I go through my talk. So this is a very simplistic overview of Antarctic climate change. So basically, if we look at Antarctica, we have 2 halves. We have one half where the peninsula is warming really rapidly, one of the most rapidly warming places on the planet. Then we have the eastern side of the continent that has tended to stay relatively cool. And we've also got ozone depletion, which over the last few decades has increased UVB radiation across Antarctica. But Probably more important, what ozone depletion and climate change have also done is pulled the westerly winds, the jet stream, closer to Antarctica. So it's actually moved in position to be closer around the continent, and also intensified in speeded up, is more intense. And I'm not an expert in that, but one of the great things about SAEF is that we have Julie Arbusser, and we have our colleagues from BOM, and we have Christina Saunders, who all have been working on these shifts in the westerly winds, and what that means and what we expect to happen in the future. What's that going to mean for precipitation and wind speeds and cloud cover and all of those things in future decades. And although I said that the Eastern Antarctica has been largely cold, this year we had an anomalous early breakdown of the ozone hole, and we ended up with heat waves across Antarctica. So we're potentially entering into space where we have this very uncertain time for Antarctica. And what does it mean when we start getting 20-degree days in Antarctica, as we saw this summer in February, breaking all previous records? So I want to introduce you to an Antarctic old growth moss forest. So this is one of the moss beds that I work on at Casey Station. And imagine that you're walking through a forest that's about the same size as my hand. So these mosses grow to about 14 or 15 centimeters tall, but they can be 500 years old. And this is some work, if you're interested in this in more detail, go back and look at Melinda Waterman's talk from a few weeks ago. So 500-year-old mosses, equivalent in my mind to a forest. As you can see, beautiful green swathes of moss, meters in diameter. And we work at Casey because it has some of the most extensive and best developed moss beds in the whole of the continent. So these 500-year-old trees have an understory of algae and fungi and bacteria. And if you were lucky enough to be walking through them, you'd be coming across tardigrades, those tiny little animals that I showed on the first slide, lots of invertebrates that are living in those moss forests. So what do these moss forests need to thrive? And like other plants, they need water, they need light, and they need nutrients. So what does that mean in Antarctica? So the most important thing is water, because Antarctica is a desert, and basically all of the water is locked away as ice. And this is some work that we've done with colleagues down at Terraluma at the University of Tasmania, using drones to map moss health across catchments. And in this image, you've got moss health. So red areas are very stressed moss, and blue areas are healthy moss. And the white over here is a snowbank, which at the moment is relatively permanent and gets replenished each winter. And the blue lines show where the water is draining, and that's some work that Diana King has done. And if you look at where the moss is healthy, it's healthiest where the water is running through the moss bed for most of the summer. So it's not just good enough to have snow. There's no precipitation, there's no rain through the summer, but it's not enough to just have precipitation. You have to have these snowbanks that are draining water through the catchment. So understanding connectivity with ice banks, understanding the future of those ice banks, and understanding how connected each piece of moss is really important to that. So we've got areas where the moss is really quite close to the snowbank, but because it's not in a drainage channel, this moss is essentially dead. And for those of you that are remote sensors, dead moss is green, dry, completely, it's not dead, it's dried, it's frozen and dried. And that will stay green and look quite happy in an NDVI signal, but actually is not active because it has no water. So you have to understand the chlorophyll signature to tell you when there's moss there, but you also have to understand if it's wet, because if it's not wet, it's not going to be active. So water is really, really important. And then the second thing that's important is, I said the mosses will need light, because obviously for a plant it's going to need light for photosynthesis. But the other thing is, how warm do these mosses get in the summer? And are they really cold climate plants? We know that Antarctica is a frozen desert, so presumably they're really good at surviving in cold conditions. And you'll notice that the colour of the moss is actually quite dark, and this enables them to absorb solar radiation and warm up in the sun. So when we go out and measure them, we have moss temperatures that are 20 degrees when the temperatures are about 1 or 2 degrees, which is the normal sort of temperature for the summer. So in that graph, you can see that on a hot sunny day, the moss beds will get to 25 to 30 degrees on a clear sunny day. And even on a cloudy day, they'll often get to 10 degrees. So they're much warmer than we might anticipate just from air temperatures. And one of the students who works with Jaume Flexer in Mallorca and who's done some work with us looked at what the temperature optimum for photosynthesis and respiration and all the other processes that govern productivity in these mosses were. And these are the temperature optima. So we get temperature optima between 19 and 26. So they're really quite like the plants we get around here in Wollongong or even Brisbane, Melbourne. Their temperature optimum is not particularly different. They just manage to survive in these microhabitats where they're able to actually achieve the temperatures, achieve 25 degrees, 30 degrees some of the time. But if we look at what proportion of the time they spend at those temperatures now, Alicia modelled that they basically have about 2.5 or 3% of their time when they're above 14 degrees. So almost all their productivity is taking place in a really small window in the summer on the days when it really does get to be above 14 degrees. So I think that's quite interesting, both in terms of what that means for how they survive now, but also what does that mean in the future? So are they going to get too hot when we have 20-degree days? Are they actually ending up being 40 degrees because they're heating up to 20 degrees above ambient? So I don't know the answer to that question, but I think it's quite interesting. And the final thing they need is nutrients, and almost all the nutrients come from marine sources. So the moss beds at Casey that we see are there because 3,000 to 8,000 years ago, there were penguins nesting on these sites. And we know that because there are tiny pebbles that the penguins use to make their nests. And you can probably see in this picture here, the stones have been graded to be a Adelie penguin nest ideal size for Adelie penguin nests. The penguins have moved, when the penguins were there, they left lots of guano. The guano has been freeze-dried for thousands of years, and now there's moss growing on it. The penguins have moved offshore, so they're now on islands off the coast, quite several kilometres away, but the nitrogen is still there, the phosphate's still there, and that's what's feeding these moss beds. And if we look across the region, the areas where we see the lush moss beds are the places where we get these nutrients from the ancient penguin colonies and the water from the snow melt. And there's one peninsula in particular that just grows lichens, and it doesn't have any evidence of these ancient penguin colonies. So it's really important to have water, nutrients, and appropriate temperatures to grow moss beds. So we've been monitoring these sites for about 20, yes, it is almost 20 years. We set up, people used to ask us what the mosses were doing in climate change or whatever else, and we didn't know. So we set up long-term monitoring in a pilot in 2000, and then we analysed it in 2003, 2008, 2011, and so on through to 2014. We were supposed to go in 2020, but because of COVID, our flights got cancelled the week before we were due to go. So we haven't actually been to these sites since 2014. But we have 2 sites and we have 30 quadrats that we've been monitoring over that time. And we do that through a number of techniques. We use vegetation percent cover from field photography of quadrats and automatic classification, which Diana King's been doing. We look at species relative abundance by taking tweezer pinches and looking at 20 little tiny shoots of moss for species ID. We do dating, which is what Melinda was talking about, to find and looking at how fast the growth rates are and looking at how old these mosses are, and using stable isotopes as climate proxies. And we, as I showed earlier, we use drones to map the area to get digital elevation models and use different sorts of sensors to pick up different sorts of spectral analyses to find out how healthy the moss is. And we've done that both from UASs and also from tripod-based sensors. So these are the photographs of the quadrats from 2003, 2008 and 2013. And Diana did automated analysis of these using eCognition to look at how much of the moss was green, how much of it was red, and how much of it was grey. And you can see that when we started off, most of the moss was green, a small amount of grey. When we went back in 2008, everything had gone red. And then in 2013, most of the moss had turned back to green again, but there was still more grey. So if we do the classification, you'll see here that there's more red and grey moss, more unhealthy moss than green in 2013 than 2003. And so this is indicating that there's been some stress to these communities. Could be drying, could be other forms of stress. And the red coloration is basically the moss is producing photoprotective pigments to protect themselves from too much energy at a time when they can't do photosynthesis very effectively. So we have 3 species that grow in the Windmill Islands. Schistidium is the Antarctic endemic, and it grows everywhere, but it grows in water as well as outside. And so the green in these pie charts is the Schistidium, and the olivey colour is Bryum pseudotriquetrum, which is a cosmopolitan species. And if we move away from the water supply, we get more of a 3rd species coming in, Ceratodon purpuris. And Jane Wasley, a long time ago, did some transplant experiments where she picked up the Ceratodon and moved it into the wet areas. And we found that you could kill Ceratodon by flooding it for 5 or 6 weeks. So we know that Ceratodon doesn't grow in flooded environments, but Bryum and Schistidium are able to cope with being completely submerged for several weeks through the summer, which is why they grow in the lakes and along the stream edges. And what we found with our species identification through the time that we've been doing this analysis is that Ceratodon, which used to be only up in those high areas, as I showed before, has moved into the lower areas that are flooded through the summer season. So from 1982 to 2000, we would only find Schistidium and Bryum and a bit of dead moss in those lower areas. But as we looked over the years, we saw more and more Ceratodon moving in. And the early work in the 80s isn't done by us. That was done by Patricia Selkirk and Rod Seppold and other colleagues who were working with the Antarctic Division in the 1980s. And they only found Bryum and Schistidium in those lower areas. So in 2008, all the moss went red, indicating that there was some stress event that had happened. But we also saw these red-shooted mosses coming in, which were Ceratodon that had moved into this area where they previously weren't found. And then in 2011 to 13, we saw about 1/3 of the plots had Ceratodon in. So it had gone from none to about 1/3 of the plots having Ceratodon in. And our only explanation for this is that that area had got drier, because that would explain why the Ceratodon had moved in all of a sudden when it hadn't been present before. So in order to test that, we looked at we dated some of these longer moss shoots, not the ones that we collected for the sampling for the quadrats. We dated them and we looked at growth rates, and we also looked at whether they had become drier or wetter based on carbon stable isotopes. And this is just to show you that the blue line is mosses that were growing in a dry area back in the 1960s, and over time their growth rate declined. And so by the 2000s they were growing a lot less fast than they had been. We had other samples that were growing in melt lakes and were actually growing very steadily through the whole time at about 0.6 of a millimetre a year. So the average growth rate for these things is about one and a bit millimetres a year. They grow really slowly. And at the bottom of the graph, we've got a shoot of moss. So basically, like a tree ring, we're going back in time and we can date it using the radiocarbon bomb pulse. And then we can use stable isotopes to tell us how wet or dry it was. And Dan Charman and colleagues who are in SAEF, who are in Exeter, have done similar work to this with much deeper cores of peat, but looking again at the growth rate over the last 100 or so years. And as I said at the beginning, the peninsula's got much warmer and wetter. So there what's happened is most of the mosses have increased their growth rates from really quite low to up to 3 millimeters a year. But you'll see in these graphs that in recent decades, there's actually been, for some of these mosses, there's been declines as well. So although over the period from 1850 to 1950, they've all increased in growth rate, in recent decades, there's been some declines even on the peninsula. So this is something that we'd like to look at in more detail in SAEF. But they do grow faster on the peninsula because it is warmer and it's probably quite a bit wetter. So the stable isotopes also enable us to look at spatially across areas and go, is this a wet area or a dry area? And so this is just showing you different environments, so dry, intermediate and wet along our transects. And we see a nice correlation between dry sites having a more negative stable isotope value and the wet sites having a more positive one. So we can actually use this method to tell us about temporal change in water availability, but also spatial change in water availability. So it's a very powerful proxy for us. And it's all determined by the fact that mosses photosynthesize, they use rubisco, the enzyme that fixes carbon, and that enzyme will discriminate against the heavier isotope of carbon dioxide if it's given a choice. But if you flood it, it takes the carbon dioxide that it's given as the carbon dioxide diffuses through the water. So the water imposes a diffusional limitation on rubisco and means we end up with different discrimination values. And we use this method to look at 18 cores across the Windmill Islands. And this is work that we did with Kwan Hu and colleagues at ANSTO. And mostly it was done by Melinda Waterman during her PhD. But when we did this, almost all the cores showed evidence of drying. 40% of them showed evidence of significant drying over since the 1960s. And only 3 of the cores showed any evidence of getting wetter, and none of those were significantly wetter. So you can see that they're just above 0. So anything that's below 0 has got drier, and above 0 it's got wetter. But you can see there's only 3 shoots that were slightly above the 0 line. So most of the Windmill Island sites that we've measured are drying. So what does this mean for these moss communities? So the Windmill Islands still has quite a bit of water where the moss beds are. But if we go to the Vestfold Hills, which is thousands of kilometres to the west, where the Australian Antarctic Division are proposing to put the new airstrip, there we have a lake called Mossel Lake, and these are pictures taken from 98 and up till 2018, showing the moss going from quite green in 1998 through to greyer and browner over the next decade. And this is what we might be, we might be in the early stages of this at Casey. So we haven't been able to study the sites at Davis nearly as much, but we would anticipate that if we looked at the isotope ratios there, we would see evidence of drying. And what we're worried about is that this might be the fate that's going to happen to the moss beds at Casey. The water, presumably the water supply at Mossel Lake has reduced much more rapidly for some reason than it has at Casey. And what's driving this? So if we look at the climate factors that are driving this, we think it's mainly due to the fact that, as I said, Antarctica's, the East Antarctic side, the eastern side of Antarctica has got slightly colder and windier as a result of this, the combined effect of the ozone hole and also the greenhouse gas and warming pushing these westerly winds closer onto Antarctica. But it's tricky to actually resolve that from the BOM record because we've only been measuring temperature and other climate parameters at Casey since the 1960s. And if you look at the graphs that I'm showing here, the main problem with it is that we've moved the station 3 times. So where we measured the climate at Casey has moved 3 times, and we've only just got to the state where we've got a 30-year record at the new station. And you can see that there's an awful lot of variability in the climate record anyway. The different colours show you the 3 different locations of the climate measurements. And so bringing it back to what biodiversity can tell us about climate, as I said, these mosses can be 500 years old. We've got a record of climate that only goes back to the 60s. So the mosses actually can tell us what's happened to coastal climate for that period where we don't have a climate record. And so I think they're a really useful potentially a really useful climate proxy that might enable us to get a better idea of what's happening to the coastal climate. And the references to the SAM, which is the Southern Annular Mode, that's the indicator that indicates the position of this westerly jet relative to Antarctica. So with a positive SAM, the jet stream is closer to further south, closer to Antarctica, and with a negative SAM, it tends to come closer towards Australia. And this has implications across the Southern Hemisphere for climate. It doesn't just affect Antarctica, it also affects us here in Australia. So climate change is drying the Windmill Islands moss beds. And so to me, this is a theme 1 affecting theme 2 connection. Is this going to continue in the future? In order to understand that, we have to understand the climate models and then understand the climate change predictions and also then scale to an appropriate level. I'm really interested in whether these effects we're seeing at Casey are much more widespread. So do they extend across East Antarctica? What's happening on the peninsula where we've got very different climate change happening? And if the region is drying, what do we do about it? Do we just watch those moss beds and wait until they've all gone brown like the stuff at Mossel Lake, or can we do something about it? So understanding, and that to me is a theme 3 issue, because it's like, could we do something, and would we be allowed to do something, and would it make sense to do something? So they're all those questions that theme 3 can help us answer. Understanding the hydrology is vital for these biodiversity health assessments, and that involves all themes, because we need to have those new methodologies of measuring hydrology and measuring where the water's actually going, and how long we're likely to be able to preserve those snowbanks. We need to understand all of those things in order to understand what's happening to hydrology. Microclimate's going to be really important for most of all of the biodiversity. And so how do we get better models of that? And again, that's all themes. And hopefully we'll be able to use mosses as an additional proxy to help us constrain the climate models. So I'm hoping that it's actually going to feed back in a big loop so that we can actually be helping to improve those climate models as well. And so that's just taking it back to that first slide in terms of, I think all 3 themes are really interconnected. I'm really excited about being able to work with all these specialists in these different areas to hopefully build those things that we would never be able to do on our own. And so that's, to me, the really exciting thing about SAEF. And I'd like to thank you all for listening. And these are all the people that actually did most of the work, not me. So thanks very much.